Hello students, today we are into zoology NEET class, and in today's class we are going to start learning about the last chapter of this unit, last concept of this unit, molecular basis of inheritance. Okay, in this concept, molecular basis of inheritance, in this chapter we are going to learn the last topic today, that is DNA fingerprinting. DNA fingerprinting. Okay, see students, we have already studied that there are nearly 3 into 10 to the power of 9 base pairs found in the human DNA. This we have already studied, right? There are nearly 3 into 10 to the power of 9 base pairs in the human DNA, right? So students, according to this, if there are 3 into 10 to the power of 9 base pairs in the human DNA, that means we have already studied one more thing, that the 99.9 percentage of the base pairs are same in all the humans. We have also studied this from the Human Genome Project. These were the observations which have been made. Human DNA contains 3 into 10 to the power of 9 base pairs, and 99.9 percentage of the base pairs are same in all the human beings. Okay, so students, 99 percentage of the DNA is same in all the human beings, then why are the human beings so different from each other? Why no two individuals are same? Yes, it is because of this 0.1 differences. Because of this 0.1 difference in the nucleotides. Okay, that 0.1 difference in the nucleotides is responsible for causing phenotypic differences in the individuals. Okay, so this remaining 0.1 percentage of differences in the base pairs, 0.1 differences in the base pairs are responsible for causing the phenotypic differences. They are responsible for causing the phenotype differences in the human beings. Okay, this is the one which is responsible for causing the phenotypic differences in the human beings. Okay, the phenotypic differences are because of this remaining 0.1 percentage of this differences in the base pairs. Okay. See students, as we have already studied understanding the genome of the human DNA, it's a very big task, right? So it took nearly 13 years for the complete identification of the base pairs in the human DNA. See students, human DNA sequencing, the entire sequencing of human DNA, it's going to be a very big work, and it is going to be very expensive also. Okay, sequencing the entire human DNA is going to be a very big job, and it requires a lot of money to be spent on it. So we cannot go with the sequencing of human DNA for every single individual, right? So we cannot go take a person's DNA and go for sequencing of every single individual's DNA. We cannot spend so much money on sequencing every single person's DNA. We have already studied what is the importance of sequencing the human DNA. It is to apply to different fields. But to apply to different fields, we have to study the DNA of each and every individual. In the Human Genome Project, we could study the human DNA in general, but in application we have to use it in person, right? So we have to apply the knowledge of the human genome. We have to study the human genome in detail completely. But with the presence of this 3 into 10 to the power of 9 base pairs, studying the DNA of each and every person is very big task. It is going to be highly cost expensive also. Okay, so for that reason this DNA fingerprinting technique has been invented. Okay, for that reason DNA fingerprinting technique has been invented. This DNA fingerprinting technique, DNA fingerprinting is the very easy and quick, cost effective method cost effective method to identify the human DNA, to identify the human DNA. Okay. So, it is not like our normal fingerprint. So, here we are going to identify DNA fingerprinting means we are going to identify the DNA of every single individual. Okay. So, it is very easy, quick and cost effective method. Okay. So, students, this technique of identifying the human DNA, this technique of identifying the human DNA called as DNA fingerprinting is mainly based on the repetitive DNA. It is mainly based on DNA fingerprinting is mainly based on the presence of repetitive DNA. It is mainly based on the presence of repetitive DNA. It is mainly based on the presence of repetitive DNA. Okay. So, what is this repetitive DNA? See students, some of the sequences of nucleotides are repeated for several times in the DNA, that is repetitive DNA. From the Human Genome Project we have identified it. Okay. So, from the Human Genome Project we have identified the presence of repetitive DNA sequences. That means some nucleotides are repeated in the human DNA. Okay. They are called as repetitive DNA. Okay. Repetitive DNA means they contain repeated sequences of nucleotides. They contain some repeated sequences of nucleotides. Okay. So, students, based on this repetitive DNA, we are going to identify the human DNA. Okay. Of any individual, of any population, we can identify the human DNA by observing this repetitive DNA. Okay. So, how do we know what is a repetitive DNA? How to identify it? Let us learn now. Okay. See students, this repetitive DNA is identified by performing some simple experiment. Like you see, we will be isolating the DNA, we will just isolate the DNA and we just fragment the DNA. That means we will be just breaking the DNA into small pieces and then we perform centrifugation. Okay. So, after performing centrifugation, we observe the amounts of DNA fragments extracted. We observe the amounts of DNA fragments extracted at different levels after centrifugation. Okay. After centrifugation, different DNA fragments will be observed at different levels. So, different fragments of DNA, produce different peaks. They produce different peaks after centrifugation if the amounts obtained are graphed. Okay, if the amount of nucleotide present in them is graphed, okay, different type of DNA, if the amount of nucleotides present in that particular DNA is graphed, then it gives different peaks when we are performing centrifugation. Okay. So, students, the bulk DNA gives the highest peak. Okay, bulk DNA produces the highest peak. Bulk DNA means the major part of the DNA which is not showing any repetitive sequences. This is going to show the highest peak. Whereas this repetitive DNA, our repetitive DNA is going to show lower peaks of different heights, lower peaks of different heights. Okay. So, students, this repetitive DNA of different lower peaks of different heights, this is called as satellite DNA. Okay. So, this satellite DNA is our basis. Entire DNA fingerprinting is based on this satellite DNA. Based on the composition of the satellite DNA, based on the length of this DNA. Okay, these are based on the size, these are based on different characters. This is categorized into different types. It is categorized into different types based on its composition or based on its size, based on the length of these fragments, okay, on this, based on the number of repeats of nucleotides, based on the number of repeats of nucleotides. Based on these categories, satellite DNA are divided into different types based on its composition, based on its size, length and number of repeats of nucleotides. Based on these different characters, the repetitive DNA, that is satellite DNA, is divided into different types. Okay. See students, example of types of satellite DNA is microsatellites, microsatellite DNA, mini satellite DNA, mini satellite DNA. Okay. So, this is a satellite DNA. So, now we have to learn what is the importance of its satellite DNA. Okay. See students, these sequences of DNA, which are repeated several times, the sequences of DNA which are repeated several times are found in the satellite DNA, right, which are repeated several times are found in the satellite DNA. So, these repeated sequences can be repeated in any number of times. They may be repeated from hundreds to thousands of times, hundreds to thousands of times. Okay. So, students, these repeated sequences are unique to every individual. Okay. That is, the satellite DNA is unique to every individual. Satellite DNA is unique to every individual. Okay. And one more thing is this satellite DNA, it can be inherited from the parents. It can be inherited from the parents. Okay. It is unique to every individual. It can be inherited from the parents. Because it is unique to every individual, because it is unique to every individual, and because it is inherited from the parents. Okay. These are the two important characters of satellite DNA. See students, one more thing here is DNA can be obtained from any cell in the body. DNA can be obtained from any cell in the body. Generally, it is obtained from the hair follicles, saliva, blood, sperms, okay, skin. In that way, from any of the cells in the body, DNA can be isolated. From them, repetitive sequences of DNA can be obtained. Okay. So, students, here, because the DNA can be obtained from hair follicles, saliva, blood, sperm, skin and any other tissue in the body, all of them, they show the same kind of repetitive sequences. All of them, they will be showing the same kind of repetitive sequences, because any cell in the body is showing same kind of repetitive sequences, and every individual's repetitive sequences are unique to them only. Because of this reason, this DNA fingerprinting will be very helpful. DNA fingerprinting will be very helpful in the case of DNA fingerprinting is going to be helpful in the case of forensic analysis. That is why we might be seeing, no students, we might be observing that in the movies or anywhere like that, we might be observing that if there is any hair follicle found or if there is any drop of blood found, that will be collected for performing some experiments in investigation of the criminal cases. So, in the same way here in forensic analysis, that is in identifying the culprits, we are going to utilize this technique DNA fingerprinting, because this DNA fingerprinting technology allows us to identify every single individual's DNA. Okay. So, that is involved in forensic analysis. So, because it is inherited, from the parents, if there are any parental issues, if there is any issues related to paternity, okay, if there is any issues related to parental issues, some people will be observing, no, that some people will claim that yes, this baby is mine, some other lady will come and claim that this baby is mine. So how are we going to identify who is the actual parent of the child? By using fingerprinting technology, by utilizing DNA fingerprinting. Yes, there is one more technique to identify who is the actual parent of the child, that is blood grouping. Yes, so the possible type of blood grouping found in the mother or the father can be only seen in the baby. Okay, from the mother and the father, the possible type of blood groups will be identified, and only that possible type of blood groups from the mother or the father can be only seen in the baby. That is one way by which we can identify the actual parent of the child. But the accurate way, very accurate way is DNA fingerprinting. In that paternity issues related to who's the actual parent of the child, in that issues also DNA fingerprinting technology is going to be very helpful. Okay, so in this way, this DNA fingerprinting technology has its applications. So students, repetitive sequences, they exhibit polymorphism. Satellite DNA exhibits polymorphism. Satellite DNA exhibits polymorphism. Satellite DNA exhibits polymorphism. What is meant by polymorphism? Variation at the genetic level is called polymorphism. Variation at the genetic level. Here in this case, variation at the genetic level is called as polymorphism. Satellite DNA exhibits polymorphism. Okay, that means the repetitive sequences are not same in all the individuals. Okay, there might be differences in the sequence that is repeated, or there might be difference in the number of times it is repeated. Okay, there might be difference in the order how it is repeated. So that is satellite DNA exhibit polymorphism. That means they exhibit some variation at the genetic level. Okay, so students, why this polymorphism exists? Why variation is occurring at the genetic level? It is because of the mutations. Polymorphism exists because of the mutations. Polymorphism exists because of the mutations. Because of mutations only polymorphism is observed. Okay, so students, what kind of mutations will result in polymorphism? See, we have already studied in our previous chapters that mutations are very common in the organisms, and mutations are resulting in nothing but there are sudden random inheritable changes. Mutations are the sudden random inheritable changes which occur in the DNA are called as mutations, right? So some mutations will result in polymorphism also. Generally students, there are two type of DNA, there are two type of genetic material found in the DNA. Okay, so one type of genetic material is the one which is exposed. One part of the DNA is the one which is expressed. Okay, the other part, one part of the DNA is the one which is not expressed. That is, expressed DNA is called as coding DNA. The non-expressed DNA is called as non-coding DNA, non-coding DNA. Okay. So students, these mutations, if they are occurring in the coding DNA, it will result in a lot of abnormalities, right? It will result in a lot of abnormalities. But if these mutations occurring in the non-coding regions of the DNA, no abnormalities may be occurring, because this DNA is anyhow non-coding. So most of the abnormalities will not occur if these mutations are occurring in the non-coding DNA. So let us learn now where these mutations are occurring, what is the reason behind the presence of this kind of polymorphism. Okay. See students, this polymorphism As I have already told you, it is due to mutations. It is due to mutations. Okay. So, students, if the mutations are occurring in the alleles, you know what is alleles? Any possible type of a gene is allele, right? So, if mutations are occurring in the alleles, then it will result in polymorphism. If the frequency of mutation, if the frequency of occurrence of a mutation is more than 0.01%, if it is more than 0.01%, then it will result in polymorphism. If the frequency of occurrence of a mutation is more than 0.01%, it will result in the polymorphism. It will result in polymorphism. Okay. So, students, mutations generally they do not occur in case of the coding DNA. Okay, mutations do not occur generally in the coding DNA, but mutations which occur in the non-coding DNA are very common. Mutations occurring in the non-coding DNA are very common. Mutations occurring in the non-coding DNA are very common. Why mutations are generally occurring in non-coding DNA? Because if there is any mutations occurring in the non-coding DNA, because it is not expressed, there will be no effect of that mutation on the individual. Okay, because this part of the DNA is not expressed in the person, there will be no harm done to that person because it is non-coding, it is not expressed in the individual. So, there is no harm to that person. Okay, that is the importance of mutations in the non-coding DNA. But students, because these mutations are occurring in the non-coding DNA, though it is not expressed, we can identify the sequences, right. So, that is why these mutations occurring in the non-coding DNA will result in polymorphism, and it will help us in DNA fingerprinting. It will help us in DNA fingerprinting. Okay. See students, this polymorphism which is which is helpful for us in DNA fingerprinting, right. So, these mutations which are occurring in the non-coding DNA, mutations which are occurring in the non-coding DNA, mutations which are occurring in the non-coding DNA, they generally get accumulated, they generally get accumulated, they generally get accumulated. Okay. So, mutations in the non-coding DNA, they generally get slowly accumulated. So, accumulation of the mutations results in accumulation of the mutations will result in speciation, that is the formation of species, formation of species, and evolution, that means formation of new variety of organisms. Okay. So, accumulation of these mutations will slowly result in the speciation and formation of, that is the formation of species and evolution. Okay. So, that is about polymorphism. So, students, no two individuals will show the similar polymorphism, okay, because they have studied now that there is a effect of, there is differences in the number of times the sequences are repeated, differences in the number of nucleotides that are repeated. Okay. So, polymorphism is different in different organisms, polymorphism occurring in different forms. Okay. Polymorphism means there is a differences in the number of nucleotides repeated, number of nucleotides which are repeated, type of nucleotides repeated, type of nucleotides which are repeated, and the number of times of nucleotides repeated. There might be differences in any of these, number of nucleotides repeated, type of nucleotides repeated, and time of nucleotides getting repeated. Okay. So, this is polymorphism, and polymorphism of the DNA, polymorphism of the repetitive sequences. Repetitive sequences exhibit polymorphism, students. Polymorphism of these repetitive sequences is the basis for DNA fingerprinting. Polymorphism of the repetitive sequences, that is the satellite DNA, polymorphism of the repetitive sequences, that is the satellite DNA, repetitive DNA is going to be the basis for DNA fingerprinting. Okay. So, let us learn now exactly what is this method, what are the steps involved in DNA fingerprinting. Okay. See students, we are going to see how this DNA fingerprinting is done. So students, DNA fingerprinting, this technique was proposed, was first developed by Alec Jeffreys. Alec Jeffreys is the person who developed this procedure DNA fingerprinting. This procedure DNA fingerprinting was developed by Alec Jeffreys. Okay. So, this technique, the DNA fingerprinting technique utilized by Alec Jeffreys involved using of DNA probes, Okay, it involved the usage of DNA probes. What are these DNA probes which are utilized for DNA fingerprinting? These are actually the variable number tandem repeats. These whatever the probes which are used for DNA fingerprinting are actually variable number tandem repeats. Okay, variable number tandem repeats, short formly called as VNTRs. Okay, variable number tandem repeats. These are the probes which are utilized for DNA fingerprinting. So these variable number tandem repeats are radio labeled and they are utilized. Radio labeled variable number tandem repeats are utilized for DNA fingerprinting. Radio labeled variable number tandem repeats are utilized for DNA fingerprinting. Okay. So, what are these variable number tandem repeats? Variable number tandem repeats are those sequences of DNA which are consisting of repetitive sequences. They are consisting of repetitive sequences. They may be varying from 0.1 kb to 20 kb length. They might be varying from 0.1 kb to 20 kb length. That means they are very small to up to 20 kb length. So, they are available in different sizes. These variable number tandem repeats contain repetitive sequences. They might be having length 0.1 kb to 20 kb also, they might be having weights. So, that means they can be very small to very big. Okay. So, variable number tandem repeats are utilized in DNA fingerprinting. The technique utilized by Alec Jeffreys, is southern blotting. Southern blotting technique. He utilized southern blotting technique using radio labeled variable number tandem repeats. Using radio labeled variable number tandem repeats. He equipped this technique. Southern blotting utilizing radio labeled variable number tandem repeats are utilized in this technique to perform DNA fingerprinting. Okay. So, let us now learn about how this exactly steps involved in the DNA fingerprinting are. Okay. So, students, the first thing we do is we isolate the DNA. That means we separate the DNA from any cell in the body. We isolate the DNA from any cell in the body. After isolating the DNA, we break the DNA into pieces. We break the DNA into small fragments by using some restriction endonucleases. Restriction endonucleases are those nucleases which are cutting the DNA into fragments. Okay, cutting the DNA into fragments. Okay, by utilizing restriction endonucleases, we are going to break the DNA into fragments by restriction endonucleases. Okay, then we perform electrophoresis. Okay, electrophoresis is that technique where current is applied. These are placed on a plate containing gel. Okay, these DNA fragments which are cut using restriction endonucleases, they will be placed on a plate containing gel. Okay, generally gel electrophoresis it is called, gel electrophoresis. Okay, they are placed on a plate containing gel. Onto this plate, this broken fragments of DNA will be transferred, and to that current will be applied. Because of this application of current, depending upon the sizes, the DNA fragments get separated. Depending upon the sizes, they get separated. Depending upon the size, these fragments of DNA get separated. These fragments of DNA, they get separated. They get separated based on the sizes. These fragments of DNA, they get separated. Okay. Then the next step is blotting. Next step is blotting. So what do we do exactly during blotting means, onto this gel plate on which the DNA fragments are separated, onto this gel plate on which this DNA fragments are separated, we place nitrocellulose or a nylon membrane. Nitrocellulose or a nylon membrane. Okay, we place a nitrocellulose or a nylon membrane, and after that we place some paper towels onto it. Okay, first we are taking an agarose, first we are taking a gel plate, we are running electrophoresis in it, then the DNA fragments are separated based on the size. After the DNA fragments are separated based on their size, depending upon their size, we are going to perform this blotting. Blotting means on this gel plate we place nylon membrane or nitrocellulose membrane on, and we will place some paper towels onto it. Okay. After we place some paper towels onto it, due to capillary pressure, what happens is the fragments of DNA get transferred. They get transferred onto this nitrocellulose or nylon membrane, onto the nitrocellulose or nylon membrane. Okay. After they are transferred onto the nitrocellulose or nylon membrane, then we do probing. Then we go for probing. Okay. What is probing? We just send the probes. We have variable number tandem repeats which are radio labeled, right? Which are radio labeled. So we have radio labeled variable number tandem repeats. We have variable number tandem repeats which are radio labeled. Now we will send these, we will pour this radio labeled variable number tandem repeats onto the nylon membrane, and we will leave them, we will give them enough time for getting hybridized. Hybridized means nothing but the DNA that we are sending and the DNA which is already existing on the nylon membrane, they will be pairing with each other. We have already familiar with this pairing. A will always pair with T, G will always pair with C, and vice versa. Right. So according to that pairing between nucleotides, when we are sending radio labeled variable number tandem repeats, hybridization of this probe DNA will occur. Hybridization of this probe DNA means attachment of this probe DNA, pairing between the probe DNA and the individual's DNA will occur. Okay. Then we will just go utilize a radio labeled film is generated. Radio labeled film is generated after this, and we observe this radio label film, and we can compare it with the radio label films of the other individuals, and we can identify what is to whom is this person belonging, or if this person is the same as that person, or who is the father of this person, or who is the mother of this person. In that way we can identify by the radio label film which is generated after DNA fingerprinting. So what did we do here, students? We have isolated the DNA from the cell, broken the DNA into fragments utilizing restriction endonucleases. After that we have performed electrophoresis, that is particularly gel electrophoresis. The DNA fragments are placed onto a gel plate, and onto it electrophoresis is done. That means current is supplied. Based on the sizes they are separated into DNA fragments or get separated. After that blotting is done. That means we place a nylon membrane or a nitrocellulose membrane onto the gel plate, and we will place some paper towels onto it. Because we are placing paper towels, due to capillary pressure, what happens? The fragments of the DNA get transferred onto the blotting membrane. Now we will send the probes, radio labeled variable number tandem repeats will be sent onto the nylon membrane or nitrocellulose membrane, and we will provide them some time for hybridization of DNA. Okay, we will give them some time for getting their DNA hybridized. After that time we will wash away the remaining probe, whatever the probe DNA which is not binding, which is not got hybridized, we will wash it away, and the radio label film will be generated which is showing the presence of different variable number tandem repeats of that particular person. We can compare it with any other DNA fingerprinting, and we can say if who is this individual, if this individual is same as that individual, or if this individual is belonging to which mother, which father, like that we can identify. Okay. So this is the importance of variable number tandem repeats and DNA fingerprinting. Okay. Let us just get take a look at one of the DNA fingerprintings. So we are taking two individuals A, B. So in the same way, like this only the radio label film will appear, and now we have the individual C. We have to see if who is the parent of this individual. Okay. So now we can identify, see this and this repeats are same at the same level in the DNA of A and C. And you can observe this and this are also at the same level, this and this are also at the same level. We are comparing C with A and B. C is not having any similarities to B, you see. C, A, same, and one is intermediate. Okay. So one is intermediate. So like this is the fingerprinting map. Okay, DNA fingerprinting map, autoradiograph, we call it as autoradiograph. Okay. So you can observe from this DNA fingerprintings of C and A and B, we say that A is the parent of C, B is showing no similarities with C, so B is not the parent, A is the parent of C. Like that we can identify the paternity or the maternity of any individual. Okay. And we can compare this DNA fingerprinting with the DNA fingerprintings of any other samples. We can identify if this person is the same person or if this person is related to this person. In that way we can identify. Okay. So this DNA fingerprinting technology has got even more advanced after the invention of another technique, polymerase chain reaction, which is short formally called PCR. polymerase chain reaction. After the invention of this method, polymerase chain reaction, DNA fingerprinting has become even more easier. Okay, after this technique, polymerase chain reaction, DNA fingerprinting technique has become even more easier. Okay, so students, in this way DNA fingerprinting is enabling us to identify the genome of an individual, to identify the genome and compare it with the other individuals. Okay, this is having its applications in DNA fingerprinting is having its application in identifying a person, identifying a culprit, identifying the maternity and paternity related issues. Okay, it is very less expensive, it takes very less time for performing the experiment, and data storage and analysis is also very easy. Instead of going and reading the entire human genome and compare of two different people and comparing them and coming to analysis, this is very easy, this is very clear pictorial representation of belongingness, of relatedness between the different individuals. Okay, so this DNA fingerprinting will also help us in understanding the evolution. It will help us to understand the mutations. Okay, it will help us to understand the variability between the individuals. Okay, so these are the various applications of DNA fingerprinting, students. With this we finished to learn DNA fingerprinting completely, and we have also finished to learn this chapter, molecular basis of inheritance. Okay, thank you all for joining the class today. We'll meet again in the next class. We will start a new chapter in the next class. Thank you all for joining.